We have seen unprecedented growth in enabling molecular level analytical technologies that have revolutionized the study of life, allowing us to measure genes, proteins, and metabolites in extreme detail in the last 50 years. In many ways, the modern life sciences resemble data science more than the study of biology in the last 200 years. Our lab studies metabolites with mass spectrometry and bioinformatics. While our universe may have started with the Big Bang, our breathable atmosphere likely originated from many gaseous eruptions from photosynthetic cyanobacteria. We now believe that microbes paved the way for animal life on our planet by emitting oxygen, and microbes sequester more CO2 than trees. Over time, we've learned to harness microbes for many beneficial things, such as bread, cheese, and wine, and alcohol. This has allowed us to live together and led to the development of civilization. And in return, we provide a habitat for many groups of bacteria to live among us. In 1676, Leeuwenhoek identified the first bacteria through a microscope, but very quickly in the 1900s, we went to war with them and started to view them as germs and eradicate them from our bodies. This led to a massive shift in human health. Lifespans doubled. My interest in microbes actually began in studying symbiotic relationships in organisms such as the Hawaiian bobtail squid, where we now know that the microbes glow to allow squids to avoid predation, and this is all done through small molecule interactions. But I'm going to diverge for a second and talk about some seminal work in the labs of Nathan Schmidt, Steve Wilhelm, and my lab that studied the malarial parasite in a group of mice that were undergoing infection from Plasmodium ueli. Now, Nathan studied the infection of these mice. I went from behind to ahead. Studied the infection of mice where he would allow them to become infected with malaria. This led to a buildup with parasite. He would study the immune system and the mice would die, leading to a dead mouse, and he would do a lot of science. Unfortunately, his breeding colony of mice got a venereal infection and he had to use a different vendor. The mice from a different vendor, Jackson Labs, turned out to be totally immune to malaria. This was great for the mice, but terrible for Nathan, or so we thought. So through some conversations with Steve Wilhelm, this crazy idea that the that the gut microbiome was responsible for the immunity came about. So the first thing we tried was looking at the diet. Turns out that a good diet allowed the mice to survive, and a bad diet, again, led to the dead mice. The next thing that we tried was to do fecal transplants, where the feces from mice that had the NCI microbiome were put into milkshakes and fed to the Jack's mouse and vice versa. That led to mice that had the NCI microbiome becoming dead mice, and the mice that had the Jack's microbiome persisting. This conclusively showed that the microbiome itself could protect the mice from malaria. What ended up happening, what we found was this was due to actually small changes in species. Most of the species, like the Muppets and the characters from Sesame Street, were very, very similar. However, some other species, like Cookie Monster and Veggie Monster, were very, very different. It turns out species in milk, gotta love your milk and hate your veggies, were responsible for the mice surviving. Now, on a more serious note, we did contact the Gates Foundation and ask, could we provide milk and dairy products to sub-Saharan Africa because yogurt was 100% sufficient to stop malaria in the mice, and it turns out that due to corruption, we could not. Think about the impact that could have had. 1,000 children die a day because of lack of dairy in Africa. That's the equivalent to our incoming first-year class every year. We now know that intrinsic factors, our sex, our genes, our environment, change our microbiome and our ability to maintain health. In fact, we are so in tune with our microbiome that from development, our microbes pattern the way we generate and progress. Our birth weights are determined by our initial inoculants of bacteria, and many molecules that are produced within us pattern health and diseases. In fact, there's a linkage of over 90% of diseases in the human body have a linkage to molecules that are produced in your gut. That is a reason to take organic chemistry, which I teach every once in a while these days, even in tissues as distant as the brain, the heart, and the liver. In a recent study that's led by Katie Morgan in the nursing college, we've been studying patients with ALS, Alzheimer's, and Parkinson's disease, and we've recently found a set of 19 molecules that are linked to specific bacterial taxa and also co-occur with all three of these neurodegenerative diseases. What's interesting about these molecules is they were previously thought to only be found in the environment, in environmental microorganisms, but we now think that this molecule, dihydroxypentane sulfadione, is responsible for being transformed into hydrogen sulfide, which is linked to stress and inflammation in the molecules. Also, I'd like to note that it comes from eating green leafy vegetables and goes through flux. Once again, veggies aren't always good. Great for me, I hate them. The work is ongoing. And I would like to point out that work is ongoing and complex. But really, the point of the talk is that as we understand the importance of the microbiome, there are many factors, extrinsic and extrinsic, that impact our human health. And more importantly, there's a lot of social factors, the cars we drive, the bears we feed, the personal care products that we use, that impact everyone's health as a collectively. And so to solve the microbiome problem and impact world health, we need to think beyond the natural sciences and work as a group to improve the world and think about conservation of the world's smallest animalcules that we have overlooked with modern medicine. So, okay.